So let me tell you something about otitis media and why it's an important disease in children and why the current therapeutic approaches are rather poor and need improving. So otitis media just means an inflammation of the middle ear. And the middle ear is this cavity between the outer ear and the inner ear. So the outer ear is where we receive sound. The middle ear cavity on the other side of the tympanic membrane has these ossicles which transmit the vibrations of the tympanic membrane to the inner ear. And it's in the inner ear, of course, where the process of auditory transduction takes place, where those sound pulses are converted to neural impulses to the brain. Now, the middle ear performs a very important function in terms of the transmission of sound, but importantly, as part of that function, it's an air-filled cavity which is connected to the nasopharynx by the eustachian tube. And when the eustachian tube opens, it equalizes pressure across the tympanic membrane. So in fact, the function of the middle ear cavity is effectively to maintain equal pressure on either side of the tympanic membrane between the outer ear and middle ear. And anybody, as I'm sure many of you have done over the last few days, will recognize that effect, that when one goes up in an airplane, if you haven't swallowed and the eustachian tube is closed, you will have not equalized pressure across the tympanic membrane, and you notice the impact upon your hearing. Your hearing becomes dampened. You effectively have a short-term conductive deafness. And indeed, if this cavity gets full of pus or fluid, as sometimes happens when you have a cold, you also notice the impact upon hearing. Your hearing is probably reduced by 20 to 30 decibels. You have a mild hearing loss while you have that particular cold. So this middle ear cavity is incredibly important in auditory transduction. Now, there are two really major types of diseases associated with inflammation in the middle ear cavity, which causes hearing loss. The first is acute otitis media, usually of a suppurative state, which is caused by a bacterial infection in the middle ear. Most children by the age of 5 will have had at least one bout of acute otitis media, where they have a bacterial infection in the middle ear. It usually clears up of its own accord, but particularly in third world countries, it's a major cause of death from a chronic suppurative infection, which then leads on to sometimes a brain infection as well. So this in fact, chronic infection affects a large number of people worldwide, a significant number of deaths. But that's not the form of otitis media that I'm really going to focus on today, because this otitis media is relatively well treatable and usually resolves of its own accord anyway. But the other form of otitis media, which is not associated with an infection, is otitis media with effusion. This is a chronic middle ear inflammatory state. And most children by the age of 5 will have had some episodes of otitis media with effusion. But in many children, as I said, it leads to a chronic state where their middle ears are full of glue, this effusion, which doesn't resolve naturally of its own accord. The children suffer prolonged hearing loss, conductive hearing loss, with all the effects that that hearing loss bring, including social isolation, learning difficulties, speech difficulties, and so on. It's the most common cause of hearing loss in the world, and the only way to treat this is by a surgical procedure. This is the current treatment, where you insert these so-called grommets, these little tubes into the tympanic membrane. It's the most common surgical operation in children. And in the United States, there will be over 2 million of these operations carried out every year at a substantial healthcare cost. The trouble with grommets is that, first of all, we don't really know why they work. We have no idea. It's kind of like 16th or 17th century medicine. It's thought maybe to relieve pressure or fluid from the middle ear. But they are relatively ineffective. They don't always work, and they tend to fall out. So children need to have them replaced, which is another surgical operation. So the current therapies are rather inappropriate. We want to, first of all, understand why does this chronic inflammatory disease in the middle ear develop, because nobody knows why it develops, what are the underlying reasons. And secondly, through that, can we develop a better alternative therapy to otitis media? If we look at otitis media, just to summarize, we can see what we call an otitis-prone condition. That is that we have different types of effusion that go all the way from a suppurative effusion, which obviously is involved with an infection, to mucus and subclinical effusions. And then, dependent upon the chronicity of inflammation, we really have two major types of otitis media. One that's generally associated with infection, the acute suppurative otitis media that I've already talked about, and the more chronic, long-lived diseases, otitis media with effusion, that are associated with an inappropriate inflammatory disease in the middle ear. But the most important point to note is that for the chronic disease, it's already known from human genetic studies that in recurrent and chronic forms of otitis media with effusion in the human population, genetic factors play a major role. So this is a disease that is really caused by changes in the underlying genes in the genome. We know that from, for example, twin studies and other human genetic studies in the human population, but no genes have been identified from these human studies so far. So we know there's a genetic basis for the chronic otitis media with effusion, but we don't know anything about what those genes are, and therefore we can't really begin to understand the molecular and cellular and physiological processes associated with that disease and begin to design new therapies. So one of the things that's come out of our screens and our phenotyping at Harwell over many years, because we've been looking for deaf mutants, generally we have a big interest in deafness genes and genes that cause deafness both in the inner ear and middle ears, that we've identified a number of mutants that cause otitis media, and the chronic type of otitis media, it's not associated with infection, that's very typical of chronic otitis media in the human population. So this is the first mutant, Jeff. It's obviously a single gene model for otitis media, and it displays a conductive deafness due to the development of a chronic otitis media with, in fact, a serous effusion. And if you compare the middle ear cavities of a wild-type mouse and the Jeff mutant here, it's a dominant mutation, you can see that the middle ear cavity is full of this pus and effusion, which is in fact causing these mice to have a significant hearing loss that we picked up in our screens. So this is our first model for otitis media. And it turns out that this is a mutation in a gene called FBOX11. Here's the mutation here. It's a non-conservative glutamine to leucine change in this particular gene. So what are FBOX genes? They're in fact specificity factors for a class of ubiquitin ligases. So they're responsible for bringing particular molecules to the proteasome for ubiquitination and degradation. And we know something, as I'll come on to describe you, about the proteins that this FBOX protein is bringing for protein degradation, but suffice to say that it's involved with turnover of proteins. The other mutation that I'll talk to you about today is the Junbo mutation. It's very similar to the mutation that the Jeff mutation. It also displays a conductive deafness due to the development of a chronic otitis media. It's a more suppurative form of chronic effusion where the effusion within the middle ear is more cellular, but nevertheless there are enormous similarities between the Junbo mutant and the Jeff mutant. Now the Junbo mutant is in fact a mutation in a transcription factor called EVI1. It's an asparagine to isoleucine mutation in this second zinc finger domain of this particular transcription factor. It's got two zinc finger domains, again in a highly conserved residue. Now, the interesting thing, to cut a very long story short, about both of these mutations, the Jeff and the Junbo mutation, is that both of these proteins, to cut a long story short, are involved with TGF-beta signaling. In the case of FBOX11, we know that it probably, through a molecule called spectrum beta 2, has an impact on SMAD3, which is involved with TGF-beta signaling. But we also know that FBOX11 is involved in nedylating p53, which is a cofactor of SMAD2 involved with TGF-beta signaling as well. So from that perspective, the other thing we know is that EVI1 is a repressor of SMAD3 in TGF-beta signaling. So both of these molecules are impacting upon TGF-beta signaling. So what has this got to do with the development of otitis media in the middle ear? So the hypothesis, and I'm cutting short a lot of work that we've done over many years, our hypothesis and our thinking was that we know that TGF-beta signaling also feeds into another pathway, and this is the pathway involved with hypoxia responses. So SMAD3 is, in fact, a repressor of prolyl hydroxylase 2, which is a molecule which is regulating the key transcription factor by which cells respond to a hypoxic environment where there's lack of oxygen. So our immediate thinking was, aha, now this middle ear that's full of pus and glue, it's not getting properly aerated, that may be part of the reason for the effect of these mutations is that the cells within the middle ear are trying to respond to the inflammatory response, but clearly the middle ear full of pus and glue is hypoxic, it's lacking oxygen, it can't be properly aerated, and therefore the response of these signaling pathways to the hypoxic environment is in fact compromised by the mutations that are impacting upon TGF-beta signaling. So I'll summarize that in the next slide just to let you digest that particular hypothesis. Our hypothesis that hypoxia is a critical determinant of the development of this chronic inflammatory state in the middle ear. We know that there's an interplay between TGF-beta and hypoxia signaling pathways, and we surmise that dysregulation of TGF-beta signaling in Junbo and Jeff impairs the response of the middle ear to the hypoxic conditions that develop. So the first question we asked, and has never really been asked before, is the middle ear in these mice, where full of pus and glue, is it hypoxic? Is hypoxia associated with chronic otitis media in these mice? And the answer is yes. We can label mice with this adjunct PIMO, which labels proteins in a hypoxic environment, and look to see whether the inner ear of our mutant mice is hypoxic. And that's indeed the case. So here's a wild-type inner ear. You can see that the epithelial lining of the middle ear is nice and clean. Here you can see the inflamed epithelial lining of the Junbo middle ear. You can also see the effusion here with macrophages and other cells within the effusion. And you can see the PIMO labeling, this brown labeling in both the epithelial lining and the effusion, showing that the environment is very hypoxic. And this hypoxia actually gets worse as the Junbo mice age and the chronic effusion develops. So the middle ear in these mice, not unexpectedly, is a hypoxic environment and is probably responding through the hypoxia pathways, the turning on of the HIF transcription factor, which is turned on to regulate genes which will deal and respond to the hypoxia that's developing. So this immediately leads us to various hypotheses and ideas of what's happening in the signaling pathways and the interactions between HIF and TGF-beta genetic pathways. So for example, we have TGF-beta signaling, which works through activating SMAD3. SMAD3 is known to be a repressor of prolyl hydroxylase 2, which is responsible for turning over HIF1 alpha in normoxic conditions and degrading HIF1 alpha. But if you have hypoxia, what happens is that prolyl hydroxylase 2 is repressed, you get expression of HIF1 alpha, and HIF1 alpha interacts with SMAD3 to increase the expression of this factor, this growth factor VEGF, which leads to angiogenesis, more blood supply, which is supposed to remediate to correct the hypoxia, bringing more oxygen, more blood to the affected region. Now you can imagine that if you have an EVI1 mutation here, this mutation in EVI1, which is a co-repressor of SMAD3, SMAD3 will go up, it will repress prolyl hydroxylase 2, we'll have more HIF1 alpha produced, which produces more VEGF, you get angiogenesis, more blood vessels, you get vascular leak through VEGF, and this is what's probably leading to the infusion into the middle ear. So this is our hypothesis of what's happening in the interaction between HIF and TGF-beta pathways. So what we did now was to go to our mutant mice to ask, is that hypothesis correct? Is the signaling through TGF-beta and its interaction with the hypoxia pathways the cause of the inflammation, the increased blood supply to the middle ear, the vascular leak, the effusion into the middle ear, the chronic otitis media with effusion, and subsequent OM. So in fact, if we look using PCR for HIF responsive genes in the ear exudates of both Junbo and Jeff mice, you can see that many of the genes that are regulated by HIF itself, but also target genes such as GLUT1 and VEGF alpha, these are all elevated significantly in the exudates of these 2 mutant mice. And in addition, we have inflammatory cytokines like IL-1 beta and TNF alpha. They're also upregulated as well. So the genes that we'd expect to see in response to the hypoxia and dysregulation of these pathways are indeed upregulated. HIF1 alpha and all of the downstream genes such as VEGF, which we surmise are contributing to the inflammation, the angiogenesis and vascular leak associated with the chronic otitis media in middle ears. And indeed, if we look at protein levels of, for example, VEGF, we find that they are indeed also elevated in middle ear fluids of Junbo and Jeff mice. You can see here in Junbo ear fluid the very high levels of both 4 weeks and 8 weeks of VEGF protein, and also in the Jeff mouse here as well. And this protein, just to re-emphasize, this is probably involved with the angiogenesis, the vascular permeability, the vascular leak into the middle ear, all of the inflammatory responses and the effusion that are associated with otitis media. We can also look further downstream. So we have HIF1 alpha upregulating VEGF, increased levels of Vegf receptor, and all of the downstream genes, which ultimately are leading to angiogenesis, vascular permeability, and so on. So we can ask if the Vegf receptor pathway genes downstream of VEGF are upregulated in Jeff and Junbo mice. And to cut a long story short, that is indeed the case. We have done VEGF RT-Q PCR signaling arrays and looked at the upregulation of genes in the exudates of both Junbo and Jeff mice. These are the data for the Junbo mutation, but we find that all sorts of downstream genes that are associated with upregulation of VEGF are upregulated in the middle ear fluids of the Junbo mice. So this is our model of what's happening in otitis media. Here we have the normal middle ear epithelial lining. We have the underlying bone, we have the mucosa, we have a normoxic environment, everything's fine. We surmise that there is some event in these mice that's kicking off an inflammatory response in our mutant mice, be it Junbo or Jeff. This is leading to an inflammation of the middle ear lining, which leads, of course, to fluid and inflammatory cells in the middle ear, to the generation of a hypoxic environment, to the upregulation of VEGF pathways. And because of the mutations that these mice carry, there's an uncontrolled cycle of inflammation, generation of fluid, hypoxia, the generation of downstream factors from the hypoxia, which are leading to angiogenesis, lymphangiogenesis, and vascular leak. So we get a runaway chronic inflammatory state in the middle ear associated with the dysregulation of the TGF-beta signaling and HIF hypoxia-responsive pathways. So that's our model, and there, as you can see, there's a lot of evidence that that's what's going on, that is the underlying molecular cause of chronic otitis media, that there are particular genes that are associated with regulating the hypoxia response that results from inflammation in the middle ear. And when that is dysregulated, you get this runaway, inappropriate chronic inflammation. But to go back to our model of how this is happening in terms of the molecular pathways, it immediately became clear to us that this provides some new target pathways for therapeutic information. Because in fact, the VEGF and Vegf receptors, which are a key component of this runaway chronic inflammation, are in fact well-known targets for a variety of cancer drugs. Many cancer drugs work on tyrosine kinase receptors, of which the Vegf receptor is one, and in fact work on the Vegf receptors because one of the things you want to do in controlling tumors is to stop the blood supply to the tumors and prevent the angiogenesis that goes on in tumorigenesis. So one of the things that we immediately looked at, because there is a very well-known class of drugs that includes drugs such as vitalinib, sorafenib, sunitinib, they are in fact targeting Vegf receptor, and we thought that if we could target this particular response that is going through, this inappropriate response that's going through the Vegf receptor, we could damp down the angiogenesis, the vascular permeability, and the vascular leak, so resolving the cycle of chronic inflammation, improving levels of fluid, improving the hypoxic environment in the middle ear, and actually treating the chronic middle ear condition in these mice. So we've taken these Vegf receptor inhibitors. We started with these three to start with to see whether we could use these to treat the chronic middle ear condition in these particular mice. So just to emphasize again, all of these three drugs, which are currently cancer drugs, our aim was to repurpose these to treat chronic middle ear condition. They act on Vegf receptor kinase. They're signaling inhibitors. They are anti-cancer drugs at the moment. Their action is anti-angiogenic and anti-lymphangiogenic in terms of trying to kill off tumors by restricting the blood supply to those tumors. There's another class of drugs that we also tried out on our mice, and that was because, of course, we know that the increased levels of VEGF are rising because of the hypoxia and the action of HIF. So we thought maybe we could target HIF signaling directly, and there's a particular compound called 17-DMAG. This inhibits a chaperone of HIF. So in fact, by inhibiting that chaperone, you destabilize HIF, and we thought we could intervene at that particular point in terms of damping down the hypoxic response and treating the chronic middle ear condition. So what did the experiments show? The proof is in the pudding. We've taken these compounds, we've treated our mutant mice with these Vegf receptor inhibitors and also the HIF inhibitor as well. And this is the experiment we've done. By 28 days, our Junbo and Jeff mice have developed a full-blown chronic disease which goes on for their complete lifetime. They never recover from it. The inflammation, in fact, gets worse. So we've carried out at 28 days a systemic administration of these drugs to either Junbo or wild-type mice as our control. And then having administered that drug to them for a whole month, at 56 days, we then test these mice for their hearing responses using a test called ABR. This is an auditory brainstem response. It just measures the level of hearing that the mice have. And we looked to see what was the change in hearing between 28 days and 56 days. In mice that are affected, the hearing tends to, in Junbo mice, the hearing tends to get worse. It certainly doesn't improve. Of course, in wild-type mice, it stays quite normal over this time period. But we're also able to look at 56 days after treatment at the histopathology of the middle ear to see, has the inflammation of the middle ear lining, has the innervation, the blood supply to the middle ear mucosa, has that changed as well, and so on. And the answer was we got a significant improvement in hearing in these mice. The Vegf receptor inhibitors moderate hearing loss for all of the drugs. I'll just take you through this slide relatively carefully. For each of the drugs, we've obviously treated wild types as control. We also did a sham treatment of the mutants. And of course, we also treated each of the mutants with the drugs. So the bar that you need to focus on are these these blue bars, and particularly compare that to the sham-treated mutant mice in the green bars. And the wild types, if you like, are our controls. You can see that in the wild type, this is the delta ABR. Here we have no change in hearing over the period. And as you can see, the wild type mice in the red bars, they're not showing any change. The sham-treated mutant mice, you can see that over the 4 weeks that we treated them, you can see that the delta ABRs are significantly positive. The hearing's getting worse as the chronic inflammation develops in the middle ear, and these mice can't hear very well. But the important and critical point is that in each of the treated mice, you can see that there's a highly significant amelioration of the hearing loss. In fact, it goes more or less back to wild-type levels, where we have a delta ABR of around 0. Of course, it varies somewhat depending on the drugs that we've used, but you can see that there was an enormous improvement in the hearing loss over that time period between 28 days and 56 days. So these drugs are actually working. They're stopping the chronic inflammation. And in fact, it's proof of principle that the underlying molecular mechanisms that we surmised from looking at the molecular pathways involved are responsible for these diseases. We can also, as I look, do the histopathology. We can look at the mucosal changes in the middle ear and inflammation. So here's a wild-type middle ear at 56 days. You can see this very clean mucoperiosteum, the mucosa, very thin mucosa. This is a totally normal mucosal epithelium. Here's the jumbo mice at 56 days. You can see this inflamed mucosal epithelium, the polyps projecting into the middle ear cavity, and this very rich cellular exudate in the middle ear cavity as well. And the question is, did we see improvements over that time period between 28 days and 56 days in the inflammatory condition of both the middle ear cavity and the mucosal epithelial lining? And the answer is yes as well. These drugs moderate mucosal proliferation as well. And again, what you need to do is compare particularly the sham-treated mice with the drug-treated mutants. You can see that there's a significant reduction in the number of blood vessels, which is obviously a measure of the mucosal proliferation in these mutant mice. So there's a significant improvement in the drug-treated mice. And also there's a significant improvement in the lymphatic vessels that usually proliferate in the inflamed mucoperiosteum. You can also see that if we compare drug and sham-treated mice for the amount of extracellular fluid in the middle ear cavity, you can see that there's also significant improvements in the number of ears with no fluid as compared to the sham-treated mice, most of which of the mice have, on these different drugs, considerable fluid in the middle ear. So we're getting an improvement in the histopathological status of these mice as well. So targeting VEGF signaling pathways in these two mouse models, Jeff and Junbo, does seem to lead to moderation of hearing loss, reducing fluid in the middle ear, reducing formation of new blood vessels. So using these compounds and hitting this particular receptor, which is key to the response of the hypoxia pathway, is improving the inflammatory downstream outputs of increased VEGF signaling. And for us, this represents a new potentially new therapeutic route for OM. And just to leap ahead a bit, our thinking immediately was if you wouldn't want to give these drugs systemically to children. These are anti-cancer drugs. But of course, the middle ear cavity is very reachable. We can get stuff across the tympanic membrane. You can, in fact, do it directly by injection. So our immediate thoughts was topical delivery of these drugs to the middle ear, moving the treatment of chronic otitis media from the surgery into primary care practice, of being able to take children into your GP, apply an appropriate drug through the outer ear into the middle ear cavity, and directly targeting the molecular changes that are going on in this inappropriate chronic inflammatory disease. So that's the way we're thinking forward, and let me just elaborate that a bit more. Because another thing that we've now hooked up with AstraZeneca, who have a number of interesting compounds which target this pathway as well. In fact, we're looking at another compound, zaracatinib, which targets one of the cofactors of Vegf receptors. And we're establishing the systemic efficacy of this drug as well. But importantly, we're evaluating ototopical delivery of the drug and plasma levels following depot injection. So what we want to do with our mouse models now is put these drugs that are targeting the Vegf receptor pathway directly into the middle ear of these mice to see whether we can, through topical local delivery, whether we can get the same results that we got with systemic delivery. And of course, if we can do that, we can begin to think of mechanisms for topical application of these drugs in children, directly treating the chronic inflammatory condition. Now, one of the questions that I'm sure you're asking in terms of going forward with this is, well, are middle ears of children who've got chronic otitis media, are there middle ear infusions? Do they look like mice as well? Is the molecular basis of the condition in humans the same as mice? And clearly, if we want to use these drugs, we have to have some confirmation that's the case. And the answer is yes, because, in fact, we've now sampled many more than 18 children. But children that are undergoing grommet surgery is an opportunity to take the effusions out of these kids' middle ear, to look at the exudate and ask the question, what's happening to changes in transcript levels and protein levels in the middle ear exudates? So in fact, we've been doing RT-PCR of all of the genes that are in the HIF-VEGF signaling pathways in middle ear fluids of children who are undergoing grommet surgery for chronic otitis media. And we find that, I haven't got all of the details here, but this is just the key finding, that the key genes, such as VEGF, the cofactors involved with it, HIF-1 alpha, many of the other genes that are involved in signaling through these pathways, just as with the mice, are upregulated in the middle ear fluids of children. So our findings mirror those that we see with mouse models, and we believe that the molecular pathways that are involved with chronic glue ear, chronic otitis media in children are the same as we see in our mouse models. So in terms of future developments along this route, what we want to do is further elaborate the mechanisms of chronic otitis media. There's an enormous opportunity here with the mutations we've got to look at additional inhibitors, small molecules that might impact upon the pathway and represent other drugs that we can repurpose for treatment in chronic otitis media. So we need to investigate that further. And of course, using the AstraZeneca drug is just the beginning of that. So we will be looking at other drugs to see how well they work and look at whether they can be repurposed for treating chronic otitis media. We need to go on and investigate additional models of chronic otitis media. I just want to say a couple of words about that, because we have a number of other mutants which show chronic otitis media, and we need to validate this whole approach. Is all otitis media resulting from this dysregulation of TGF-beta signaling and its impact upon HIF signaling in the hypoxic conditions that generate? And the answer seems to be yes, at least for one other mutation that we have. So I've talked to you about the Junbo and Jeff mutations. We also have another otitis media mutation called Edison, which is a mutation in a gene called Nisharin, which we don't really know what it does. This is one of these orphan genes that we don't really know much about. But we have a paper in press of another mutation, which is this a gene called TGIF, which is involved with the TGF-beta pathway and also causes otitis media in the mutant mice. This gene, TGIF, is one of the regulatory molecules involved with TGF-beta signaling. It's called transforming growth inhibitory factor 1. And I won't go into all of the details. I'll just point you to a couple of panels down here. It develops a chronic otitis media very similar to Junbo and Jeff. So it's another gene in the TGF-beta pathway which is causing chronic otitis media. So do go and have a look at that paper if you're interested. So we're beginning to get more mouse mutations which are validating our thinking on the key genes that are involved with leading to chronic otitis media. So, of course, we've got to look at these additional mouse models. We're investigating the underlying molecular basis of human chronic otitis media and building on the LOOM study that I've told you about. And indeed, our findings are that the molecular basis in humans is very similar to the mouse. And of course, last but not least, if we want to get these drugs into the middle ear, we've got to think more about the delivery systems, and the mouse can be a test bed for this. We need to think about how do we get those compounds across the tympanic membrane into the middle ear. And we're looking at various technologies that make the tympanic membrane more permeable to drugs, all the way from patches that you put on the tympanic membrane to compounds that you can use to make the tympanic membrane more permeable, and you could use in eardrops that you put into a children's outer ear. But the whole goal here is to move the treatment of this condition into primary care, that you simply go along to your primary practice nurse who delivers a drug to the kids, maybe recurrently once a month or something like that, to damp down the chronic middle ear inflammation. So the conclusions here are we've found that Jeff and Junbo are models for human otitis media carrying mutations in these two genes. And we've shown that FBOX and EVI1 cause otitis media through effects on signaling within the TGF-beta pathway. They provide a common mechanistic route for development of chronic otitis media that involves dysregulation of hypoxic responses in the middle ear. We've shown that transcriptional profiling and the effects of Vegf receptor inhibitors support the role of hypoxia signaling pathways. And clearly, treating these targeting these pathways represents a novel therapeutic avenue for new clinical applications in the treatment of chronic otitis media. And one slide that I actually forgot to put in here, but I'll just tell you now, is that, of course, and we have done this, we can go to the human population. We have thousands of chronic otitis media children out there, and we can analyze their DNA to see whether they're carrying variants in some of these genes that are involved with TGF-beta and HIF signaling. And to cut a long story short, and I forgot to include the slide, I should have included it at the end because it's a nice ending to this, is that one of the key genes that's often associated shows strong, significant associations in the human population with the development of chronic otitis media is FBOX11. This has been found in a number of human association studies. So we have both genetic evidence as well as evidence from the LOOM study of studying effusions in children with chronic glue ear. We have genetic evidence as well that are thinking about the role of these pathways in the development of chronic otitis media is indeed correct. So it only remains for me to thank all of the people who are involved in this work at Harwell. This is the otitis media group and the people who have been involved over the years. And this work is down very much to two people, Michael Cheeseman and Hayley Tower, who've done much of the work that I've talked about today. And thank you for your attention. Thank you. Thank you.